So I guess we'll get started. Thank you all so much for joining. My name is Bill. I am the lead for the GDC User Services team, and today I'd like to talk to you about genomic mutation analysis in GDC 2.0. I'd like to show some of the features and things that GDC 2.0 can do. Moving to the agenda. So before I get started, I'd like to say that we're going to answer questions from the audience at the end. So if you have any questions during the meeting, just feel free to put them in the Q&A panel, which is sometimes you can't see it right away. There's 3 dots, and when you click on it, there's a Q&A option there. So I'd recommend clicking that. We might not see what's in the chat, so make sure you get your question in the Q&A. So the beginning of this webinar is going to be an overview of genomic mutations in the GDC, just to give you an introduction, followed by a kind of a recap of creating custom cohorts in GDC 2.0, because this is a cohort-based framework, and mutation analysis is no different. We will be creating a cohort. After that, we'll demonstrate 3 tools, 3 analysis tools from the GDC: mutation frequency, OncoMatrix, and protein paint. If we have some time at the end, we're going to go over how to download genomic mutation files, specifically how to query them. And then at the end, we'll take any questions from the audience. So starting out with genomic mutations in the GDC, mostly we're talking about somatic mutations, and there's 2 types we're talking about. The first type is simple somatic mutations. These include SNPs and indels. So here's the basic steps to how this is created. We have other webinars that go a lot deeper into this, which I'm going to mention in a bit, but the first step is that whole exome sequencing and targeted sequencing, tumor and normal reads, are aligned to the GRCH38 reference genome. These are 2 separate BAMs, and these are made available in the GDC data portal. After the BAMs are compared with, they're run through 4 different pipelines to generate variant calls. That would be MUTEC2, Muse, Verscan, and Pindel. The first 3 being SNPs, and the last one being small indels. So this creates 4 VCF files. After that, the variant calls are annotated. This is just adding more information to each of the calls, and these are available in VCF and MAF format, 4 of each, one for each pipeline. Lastly, and most importantly for our case, the variant calls are aggregated and filtered. So this is this results in 2 separate MAFs, one open access and one controlled access. And I want to mention that when I say aggregated, I mean that for a single tumor normal pair, you have 4 different pipelines, and all 4 of those are aggregated into one file, one MAF file. And that MAF file is controlled access. It is put through a filtering pipeline in which anything that has a chance of not being somatic and could be germline is removed, and that results in the open access MAF. If you're interested in the specifics of that filtering, I'd recommend watching the September 2020 webinar on GDC DNA-seq data processing. Some GDC bioinformaticians give a very good explanation on how to do that and how that's done. So I'd recommend checking that out. Once those open access maps are ready, the variants from them are indexed in the GDC API and made available through that and through the portal, which we're going to show during our demo. So the second type of mutation are the copy number variation mutations. These are created using 2 different pipelines in the GDC. So the first one is the whole genome sequencing pipeline. These are aligned the same way that whole exome is aligned. Tumor and normal are compared, and then gene level copy number variation calls are generated with the ASCAT NGS pipeline. The alternative way that they're generated is in some of our older data sets. The one you'll see today is TCGA. They have copy number variation calls from the SNP6 array. This one has two more normal pairs. Those are used to generate CNV calls with the ASCAT2 pipeline. After either of these is generated, the gene level CNV calls are indexed in the GDC API the same way the simple somatic mutation ones are. So yeah, let's get on to the actual data portal tools. So these are the three that we're going to go over today. The first one is mutation frequency. This allows you to summarize the mutations in your cohort, visualize survival in mutated versus non-mutated, and this is at a gene or mutation level, or filter and create sets of genes or mutations based on what your requirements are. The second tool is OncoMatrix. This is a matrix, as the name suggests, that visualizes mutations and cases in a matrix by genes. This also allows you to visualize the mutated cases alongside clinical attributes. So these can be added to the visualization, which you're going to see later. Lastly, Protein Paint is a tool that allows you to visualize mutations on a polypeptide chain and some other chains that we'll show you in a bit, and also display mutations on the full genome. Just to reiterate, maybe you saw the last webinar on GDC 2.0, but this is going to use a similar workflow, so I just want to reiterate how that goes. This is the main workflow, but today we're only going to really use two parts of it. The first step is to build a cohort based on whatever attributes we're looking at. This will be a group of cases, and everything that we bring the cases to will only apply to those cases. So if we look at the mutations in this cohort, it'll only be for this cohort. The second step is to use the cohort with whatever tools we're looking at. And here I've circled in magenta the three tools that we will be looking at today. These will be automatically applied to the cohort, and if you switch between the tools, it'll be the same cohort unless you switch the cohort. So let's get on with that and start with the demo. So I'm going to go ahead and end the PowerPoint real quick. And once I minimize this, you should see on the left here are some slides that I use to keep myself organized. And on the right is the GDC data portal. So we get here by going to portal.gdc.cancer.gov. Warning will come up about, just making sure you're using it correctly. And so here we're at the main portal page, and we want to build a cohort. So of course we go to Cohort Builder, which is the third from the left. Click here, and the way that I've organized this presentation is that each of the tools will have a set of small goals, and these goals will be used to show you all of the features in GDC 2.0 for genomic mutations. So the first one is to create two cohorts for mutation analysis. These, by the way, this doesn't have any clinical relevance. It was just sort of randomly chosen on my end, so just keep that in mind. So we're going to go ahead and create our first cohort, which of course is program TCGA. So we'll go to program, click on the search values, and type TCGA. Here's the value here, so we'll go ahead and click that. And as you can see, the number of cases at the top, it used to be around 44,000. Now it's going to update to be around 11,500. So next, we're going to filter down for any case that has a tissue or organ of origin that has a value of upper lobe lung. So let's go ahead and search for just lung so we can see all of our options. And here, down here, we have upper lobe, so we'll go ahead and click that. This has 563 cases. And if you look in this query toolbar here, you'll see that all of the different attributes that are selected for this cohort are are displayed here. So these can be like collapsed or made smaller, but we'll keep it visible for now. So we'll click the save icon here, and we will save it as upper lung. We'll go ahead and click save. And now, as you can see, upper lung is our active cohort. Now, our next cohort is the same program, TCGA, but we want a tissue or organ of origin of lower lobe lung. So we don't have to create a new cohort from scratch. We can go ahead and use this one and just kind of edit it in place. We'll click on lower, we'll unclick upper, and now it says changes not saved. So we don't want to write over upper lung because that is a cohort we're going to use. So we'll go ahead and click on save and save as. This works the same as something like Microsoft Excel, in which another copy is generated while retaining the original. So we'll just change this to lower, click save, and now our goal is met. We have our two cohorts. So let's go back to upper lung, and now we're going to get on to demoing the three mutation analysis tools: mutation frequency, OncoMatrix, protein paint. So to get to mutation frequency, we're first going to click on the analysis center, which will display all of our different analysis tools, and we'll click on mutation frequency at the right here. So I'll click the play button. And before I get into what we're going to do with our cohort, I want to introduce you to the layout of the tool, just so you know what's here. So at the top, you're going to see the top 20 most frequently mutated genes here. These, as everything does in GDC 2.0, apply to your cohort. So this is not GDC wide. This applies to these 563 cases that are in your active cohort. On the right here is a survival plot that compares the two groups that are separated into not mutated for a particular gene versus mutated for a particular gene. And we're going to show you how to select that gene in a second here. Below these two graphics are a table of all of the genes that are mutated in your cohort. Now, this might look like a small number, 715 genes, and that's because we have this filter that's selected by default, is cancer gene census. And this is a pre-curated list of genes that are known to affect cancer. So this is selected. If we unselect it, you'll see that the number of genes gets much, much higher, or 20,000. So we'll go back just to make it a little faster. At the top here, you can select between looking at genes. So each gene represents a gene in a case that has a mutation in it. So even if, these 387 cases that have a gene in TP53, it might not be the same mutation. So let's go ahead and click on mutations, and the graphics not that different, except that we don't have the top mutated genes because that wouldn't work in this case. And this is a list of all of the mutations. And of course, it's much higher because you could have multiple mutations in a gene. So let's go back to genes here, and let's look at our first goal. It's to create a smaller cohort of patients who have the most frequently occurring mutation in the original cohort. So if we want to see the most frequently mutated or the most frequently occurring mutation, not the most frequently mutated gene, we'll click on mutation here, mutations. And at the top here, you can see that this actually isn't the top most mutated gene, which is TP53. It's the KRAS. It's a mutation in KRAS, which has the highest number of occurrences in this cohort, which is 24. So to save this as a cohort, we want these 24 cases. We can either click here to save a cohort, or we can click on this button here. So if we were to click on this toggle switch, you'll see at the top query bar, we now have another property added to the cohort, making it much smaller, 24 cases. So we can go ahead and save as, and we'll save it as upper lung KRAS mutation. So let's switch back to the lower lung in this case, and let's go to our second goal. Our next one is to see which cancer genome, cancer gene census genes are most commonly mutated with high VEP impacts in each cohort. And then we want to compare survival curves in the second most mutated gene. So because we're talking about genes, we're going to go back to the genes selection here, scroll down, and on the left here, we have the ability to filter for different attributes in the genes or in the mutations. So, for example, protein coding applies to a gene, and impact scores apply to mutations. So, looking at our filters, we have is cancer gene census? Yes, so we will keep this active. And then under VEP impact, we will click high. So now this is our list of genes. It's smaller, 311. And we want to take the second most mutated gene and see what the survival is between those with the mutation in the gene versus those without. So clicking on the survival button will show this. And it also creates a p-value, which shows you kind of the significance of the difference between the two. 0.67 is not a very low p-value, so there's not much difference between the survival in these two sets of cases, but that's for statistical analysis anyways. We can also go down to upper lung and do the same thing. Search for VEP impact high, keep this active, and as you can see, CSM D3, and this one actually doesn't have enough data, so we won't be able to look at this particular one. So let's go ahead to our third goal, which is to see how many cases have mutated cancer gene census genes that are unique to their cohort. So between the two cohorts, which genes are uniquely mutated? So let's just reset everything in the mutation frequency app. So we'll close it and open it. And as you can see, all of the different filters are back to their original their default settings. So let's first look here in this table. This right here is the list of the genes that have a mutation in the upper lobe lung. So let's take this list and save it as a new gene set. So go ahead and save. And hold on one second. Let's switch between lower lung and upper lung just to reset it. So going down to this list, we'll save this 697 genes as a set. So we'll save as a new gene set, and let's just save it as upper lung genes. So we'll go ahead and save. Now let's go to lower lung. And we're going to save another set of these genes, 711 genes. And we'll save that as a new gene set. And we'll call that lower lung genes. Now we'll save. And if you're interested in where those sets go, you can go to the top right here and click on Manage Sets, which shows you a list of each. And if you'd like to click on it and look, this doesn't do us any good because it's a huge list. But if you maybe had a few, this would be a good way to see it at a glance. So now that we have each of these sets, we want to see which ones are unique to each. So let's go ahead and go to Analysis Center. And I said we were going to only look at 3 tools today, but we're actually going to look at a 4th, which is set operations. And this will allow us to see which which genes are unique to each of these sets. So the sets can be genes, they can be mutations, or you can also compare cohorts in this tool. So we're going to click on the two gene sets that we created, the upper lung genes and the lower lung genes. And we're going to go ahead and run this. And as you can see, we have 15 that are unique to S1, which are the lower lung genes, and 1 that is unique to S2, which are the upper lung genes. So we're going to take these 15 genes and here is the line that corresponds to the 15 genes. And we want to go ahead and save this as a new set. So we're going to click on this button and call this unique to lower. Save it. And we'll just leave the 1 for now. So let's go back to the Analysis Center, look at mutation frequency, and now we only want to look at the genes that are unique to the lower lung. Right now we're looking at 711 genes, and that's way too many. So we can add gene filters that'll only display a subset of genes on this page. So let's click on Add Custom Gene Filters. And if we had like a list of gene identifiers from a publication or from some file, we could paste them in here. But we don't have to do that. We can go over to the tab called Saved Sets, and we can click on Unique to Lower. So I'll click on that. I'll click Submit. And as you can see, we have this list of genes here that are only applied to this set. So now this table only has these 15 genes, and these 2 graphics only apply to those specific genes. And everything that I'm saying, of course, is all within these specific cases. So the next thing we're going to do is talk about the OncoMatrix tool. So let's get out of mutation frequency and let's click on OncoMatrix, which is on the right here. As always, I want to mention that these cards won't always be in the same exact place depending on your screen size. So just keep an eye out for that. And right now what we're doing is we're loading up the mutated cases in the lower lung cohort to display here. So we have this default set of cases. Each row is a gene, each column is a specific case, and the colors represent a mutation occurrence. So the long ones that are like the entire cell is a copy number loss, green, or gain, pink. And then the smaller ones between it are the simple somatic mutation. So these are SNPs or indels. At the top are a bunch of options to customize the appearance or zoom in. So we're going to show that in a second when we have our goal, which is to visualize cohorts in one matrix with lung cancer survival genes, I'll get to that in a second, along with the case's gender and primary diagnosis. So let's go ahead and first customize our set of genes. So if you wanted to customize your set of cases, you would just change your cohort, and that would be pretty standard. But to customize a set of genes, you would click on the number of genes here, and click on Edit Group. Now this is the default set based on the mutated genes in your cohort. So we'll go ahead and clear these out, And we want to look at lung cancer survival genes. And the way we can do that, there is a pre-curated list of genes from MSigDB that relate to lung cancer survival. So we'll go ahead and type in lung here, and we'll use this Chen lung cancer survival set of genes. And this automatically populates a set of genes here. We'll go ahead and click submit. And the matrix loads. So now because we're not looking at the top mutated genes, you can see the mutations are actually a lot more sparse. At least the SNPs are. Now we want to add the clinical attributes to this matrix. And we can do this by going to variables here and choosing first the primary diagnosis. So we'll click on diagnoses. We'll scroll down to P for primary diagnosis and submit that. And as you can see, it's a bit more zoomed in now, but each of the primary diagnosis has a color that corresponds to the legends down here. So orange is adenocarcinoma and not otherwise specified. Papillary adenocarcinoma is brown here. So all of these are labeled under primary diagnosis. Um, Additionally, we can add demographic, gender, and these will be labeled with whether the cases are labeled as male or female under the gender property. So here we go. Orange is male and blue is female. So that is what I wanted to show you with OncoMatrix. And lastly, we're going to look at protein paint. So let's go to the Analysis Center. We are here, and click on Protein Paint, which at the bottom left here, here it is. We'll click on the play button, and we'll actually go back and we'll do the demo to show you the tool layout. So it first prompts you to enter a gene of interest, which I showed you before, and it allows you to kind of change it on the fly here. But looking down here, we have the polypeptide chain. Each of these lollipops here is a mutation from your cohort. This is the demo, so this isn't actually from our cohort. And then if you click on a mutation, it'll cause the lollipop to move to a more prominent position. And the number represents the number of cases that are affected by the mutation. So if I click on 2 here, you'll see that a list of 2 different patients comes up. The shaded parts on the polypeptide chain are protein domains. These are curated by external databases like CDD or PFAM. And if you had a domain of your own or would like to highlight a part of the polypeptide chain, you can add one using this custom tool here. So the other thing I wanted to show you was that if you click on each of these here, you have the option for a particular sample or case to display a disco plot. And what the disco plot does is shows the mutations in the entire genome. You can see chromosomes 1 through 22 in X and Y. And right now it's only showing mutations for cancer gene census. If we unclick that, you'll see that we have a lot more mutations. So we'll go ahead and reclick that. And we'll go back to our goal here, is to visualize missense mutations on the SSX1 gene and the full genome of one case with mutations. So let's go ahead and type SX1. We'll click on that. And on lower lung, here we have some samples. So we can We only want to visualize the missense mutations. So here we'll click on missense, we'll click on show only, and this will make all of the other ones disappear. So now blue is missense, you only see blue ones here. Now, if we click on one of these mutations, we can, of course, look at the disco plot. And once we click on that, we'll see all of the different mutations, including SSX1, in this case. Lastly, I want to show you that right now we're looking at the polypeptide view, which is kind of the default view of this gene. So if we click on this label up here, we can look at, protein is what's selected, the polypeptide. We can look at the exon only, the splicing RNA, or genomic. And genomic, it's, of course, you won't see things very fine scale because there's huge, huge introns that you won't be able to see any mutations on. But if you'd like to see how the exons are spaced out, that can be done here. So the last thing I want to demonstrate is downloading the mutation files. I'm not going to show you exactly how to download, how to perform the action of downloading. There's quite a few um, webinars that do show you how to do that, including the last one. But I'm going to show you how to get to the particular files that these mutations all come from. So let's go to the Analysis Center, or we can click on Repository here or up here. So I'll click on Repository, and this right here, these 20,000 files are the ones that apply to my cohort, the 354 cases. So if you want to see where the simple somatic mutations come from, let's say you want to download the data, you want to store it locally on your computer, and you want to perform some analysis that perhaps we don't have or that the API can't do automatically. So you can download these files by going to the masked somatic mutations, which are under data type. You can either click 18 more or just type masked here. Masked somatic mutation. So once I click on that, you'll see that the data format is MAF. All of the access is open. And they're mostly going to be open access. There's a few projects that don't have them open access just because of policies. But generally, if we can, we will make these mass somatic mutation files open access. So all you do is add them to your cart and download them. It's only 30 megabytes right now, so that would be easy to download from a browser. Alternatively, you can download a manifest and use the GDC data transfer tool. So let's unselect mask somatic mutation. And let's talk about copy number variation. So the copy number variation are two different pipelines, and you can get to those in the workflow type field by searching for ASCAT. And the ASCAT2 files, these are the ones that are from the SNP6 platform. These are downloadable here. You would want to, and we have data type filtered here. You'd want to download the gene level copy number files from this one. Alternatively, if you wanted to use the whole genome sequencing called files, you go to ASCAT NGS, and it's the same data type, gene level copy number. These files are the ones that provide the CNV mutations to the API. These are the ones that are indexed. So that's the end of the demo, but I'd like to show you around just a few of the features we have for support. So first, if you're interested in any of the tools, the ones I just showed you, and the ones that are all of the other tools in the GDC, we have video guides to all of them, which you can reach by clicking here. And this gives you a list. They're all on YouTube. You can watch them on YouTube, or you can go to the YouTube playlist, which has a list of all of them. Um, the GDC users guides can be found at docs.gdc.cancer.gov. This is where we are now. I'll click on the home page, and this just has a user guide to each and every one of the GDC features. So if you were to click on Data Portal, you will have a list of all of the different analysis tools and core tools that are available in GDC. Lastly, the GDC website, you can go to gdc.cancer.gov. This is kind of a higher level information, so you won't find user guides here, but there's a few things that users will find of interest. One is by going to Access Data, you can download the GDC data transfer tool here. Also, if you're interested in watching other previous webinars or signing up for the next webinar, you can click on support, go to GDC webinars, and that will bring you to a list of them. Then, if you're interested in knowing when new GDC releases happen, you can join the GDC user listserv, which is here. And this will allow you to you sign up, and every time there's a GDC release or something notable like a maintenance window, you'll receive a notification. So if you're interested, I recommend joining the GDC user listserv. Going back to the presentation, I just have a couple more things to show you. We have some new GDC data portal features that are coming soon that we'd like to bring to your attention. So first, we have a new tool that will be coming out called the cohort map tool. The map files that I was showing you before, the open access one, this applies to them directly, and this will automatically aggregate them based on your cohort. So there'd be no need to do that through a script or anything. This will be done automatically. And this works on open access files. So if you're interested in downloading the data that populates the SNP portion of the SNP and indel portion of the features I just showed you now, this is a good option. We have enhanced OncoMatrix features that are coming, new gene expression clustering filters, and we're bringing the annotation browser and the sample property filtering to the new GDC data portal. So GDC 2.0 just came out, and we're bringing these features to that one. There are plenty more features that are coming out too, and these will be listed in our release notes, but also in an announcement that'll be going out to the listserv. So if you're interested in the next GDC data portal release, please sign up for the listserv. And with that, we will take any questions. So let me go ahead and check that out.
Okay, Bill, there's some questions in the Q&A and some questions in the chat as well. If everybody could put their questions in the Q&A, that would be great, but we'll go through both. First, is it possible to look at a distribution of mutations for a disease and identify where a gene fails in that mutation distribution? So for example, TP53 would be on the upper end of the distribution as one of the most mutated genes.
Oh, so we do have, the top mutated genes automatically visualized on the for your cohort. So you can look at that. If there's a more specific type of distribution that you'd like to make, I would recommend downloading the mutations, locally and using a script or whatever else you'd use to do that. So going to the next question. Some map files have no data in them, just the headers. Does that mean there are no mutations between tumor and normal? And that's a very good question. So going back to actually one of the first slides, step 4 here, the variant calls are aggregated and filtered. So they're aggregated, that creates the aggregated somatic mutation file, and filtered, that creates the mass somatic mutation file. Aggregated the aggregated semantic mutation file is generally going to have some mutations in it, but this is controlled access. So when the filtering happens, sometimes that will filter out all of the different mutations, all of the mutations. So when you go to download the MAF file, you'll just click on it and you'll get a header. Now, you might say, okay, that's why would you have a file with just a header in it? And the reason we do that is just so that users know we didn't forget to include the file. It's not missing. It's just everything was filtered out. Um, if you if this is something you run into, something I recommend doing, a nice trick, is to filter your files down to a certain list and then sort it by file size. And you'll see that the header ones are always like very close in size, and then when you actually start to get more mutations, you'll see the size jump. So if you sort them and are only interested in the files with mutations in them, you can see pretty easily through the size. Um, so going on to the next question, um, is it only a web tool or is there an R package for these tools? Um, So as far as I know, this is a web tool. Um, I don't know, Sharon, do you know if there are any R packages that do these tools or anything similar? I am not sure.
Yeah, I mean, I think, for the most part, behind these tools are the GDC API. And so, you may not have, an R package to actually do some of the visualizations or tool, but we have the GDC API. There's like a bioconductor package that uses the API. So like OncoMatrix uses the GDC SSM API, and so does Protein Paint. So those are available through the GDC API, or like bioconductor has a custom GDC package.
Sharon, I was going to also say OncoMatrix and Protein Paint are open source packages that from St. Jude, so you can look into that yourself.
Okay, so looking at the chat, I see some more ones. So is there a possibility to download these mutations in a table instead of a plot and to see the gene IDs? And yeah, at the um let me go to the website. Going back to the analysis center, mutation frequency. You can see it down here. We have the ability to download as a TSV, or you could save the gene set here, so that can be downloaded. So the second one, does GDC data include the data set from Genie? And right now, Genie is not in the GDC. The GDC data portal does not have the data from Genie. I know there's some other places that do host that, so that Genie data can be retrieved from other places. And that's it for the chat. And I don't see any more questions in Q&A. Does anyone have any additional questions? Okay. With that, thank you all so much for joining and listening to this webinar on genomic mutations in the GDC. We hope you have a great.
So sorry, Bill. There's one more question that just
Okay.
popped up.
Let's see.
Seeing it's about controlled access data. I just saw it.
Oh, yes. What is the criteria for controlled access data? That is a very good question, and I am happy to answer that. So if you go to repository here and you go to the bottom, you see that there's access controlled and open. So controlled data is usually data that has genomic sequence in it. It's data that has um, real DNA or RNA sequences that can be used to identify a particular case, which is something that that we don't want and you're not supposed to do with the data. So when you do get dbGaP access, which is required to access controlled data, you write a you write a proposal and you make sure that you sign a bunch of things that say you're not going to do that. If you look at the other controlled things here, so I'll click on controlled, and we have many different things, but one of the things you'll see are the VCFs, the raw variant calls. These are the variant calls that are that come directly out of the pipelines, and this data is not filtered at all. We only use semantic variant callers. However, semantic variant callers aren't perfect, so sometimes some of their calls could be germline, which could allow one to identify an individual. So for that's the reason we perform the filtering that results in the open access maps. So we apply some very conservative filters that say, these have a very, very, very high chance of being somatic mutations and not germline mutations. So that is the basic criteria of controlled access data. Occasionally, there are projects with policies that require that data is controlled access, even if normally it would be something the GDC would make open access. But that is not something I can talk very much about due to I don't have much knowledge on that.
Okay, and it looks like a question is, what is cancer gene census?
Yeah, so my understanding is that it is a curated set of genes that that have been experimentally shown to be involved with cancer. I'd recommend they have lots of information online. So it comes from an external database. They can tell you more about themselves than I can, but we do use their curated list to filter down to the reason we use them is that so when we go to mutation frequency, we don't get these don't get kind of thrown down the lines because we have much bigger genes or things like that. So we filter this down to genes that are known to be cancer related because GDC, of course, is a cancer genomics platform. So we that's why this is automatically filtered. But if you don't really care for that, you can always turn that off and look at the full set of genes.
And the source is COSMIC, the catalog of somatic mutations in cancer. So if you go to Sanger, cancer.sanger.ac.uk/census, you'll see the COSMIC catalog of somatic mutations in cancer. So that's where it's coming from.
Ooh, how do I filter a case by case ID? So this is something you can do by going to the cohort builder. So we could start out from scratch, but let's pull a case ID out just for fun. So we'll go to our cases and table view, and let's say case ID here is TCGA 947943. So I'm going to go ahead and copy that. And sorry, some of these WebEx windows are getting a little in the way. So we'll click on cohort builder and we'll start with a we'll minimize that. We'll start with a brand new cohort. So let's click plus for a new unsaved cohort. And now we have all of the cases in the GDC. All 44,637. So if I wanted to look at one case, I can go to import new cohort and just paste it right here. As you can see, it matched one to here. It's known if I was to put some nonsense here, like not a case, it would unmatch this one because it doesn't exist in the GDC. So I go ahead and click submit. And we'll just call this one case. So now I'm filtered down to only one case. So if you'd like to do a bunch of stuff around the GDC, you could do that. Alternatively, if you were just interested in, say, the files or something, you could go and click on the case ID here. And this will give you information about, what files are available, what experimental strategies are available, the clinical information, the clinical files associated with it, and the biospecimen information. And something I didn't mention because there's just so many changes, but in the next release, there's going to be a little bit of a reorganization here to make things more useful for those seeking files. So if you're interested in one particular case, after the next software release, this will become easier. All right, let's see. Next one, is it possible to look at the changes between primary and then a refractory mutation, or the difference to see evolution and potentially see what therapy might have been used? So what this sounds like is if you were to have different time points on a BAM, or if you had a bunch of different mutation calls at various time points, this is something you would be able to see. We don't have all that much of this data right now, but and we also don't have any specific tools that look for this this specifically. So it wouldn't be visualized as like a time point or anything. But if you were to have different if you were to have multiple variant calls at different time points, that is definitely something you would be able to see if you were looking at the somatic mutations. All right, I'll just give you a minute to if there's any additional questions. Okay, well, if there aren't any more questions, thank you all so much for joining and for listening to this webinar. And yeah, I hope you all have a good rest of your day. See you later.